Hello friends. Welcome to my channel, Smart Biology. Today our topic is recombinant DNA technology. Recombinant DNA technology. After studying this tutorial of recombinant DNA technology, we will learn about the introduction of recombinant DNA. What are the application of recombinant DNA? And what are the steps involved in recombinant DNA technologies? And conclusion. Introduction. Introduction of this tutorial includes definition of recombinant DNA technology. Purpose of recombinant DNA technology. Recombinant DNA technology can be defined as recombinant DNA technology is a technique used in genetic engineering to combine DNA molecules from different sources to create new genetic combinations that may not exist naturally. Purpose of recombinant DNA technology. The purpose of recombinant DNA technology is to manipulate DNA in order to create new genetic combinations that can be used for a variety of applications, including the production of genetically modified organisms, GMOs, the development of new medical treatments, and the creation of vaccines. Applications. The application of recombinant DNA technology are GMOs, medical treatments e.g., gene therapy, vaccines. Number 1. GMOs. Recombinant DNA technology is used to produce genetically modified crops that are resistant to pests, have improved nutrient profiles, or produce higher yields. GMOs are also used in the production of medicines and industrial enzymes. Medical treatments e.g., gene therapy. Recombinant DNA technology is used to develop new medical treatments, such as gene therapy, which involves the introduction of normal genes into cells to treat genetic disorders. Recombinant DNA technology is used to produce vaccines by introducing genetic material from a pathogen into a vector, such as a virus or bacterium, which is then used to produce an immune response in the body. The step-by-step process of recombinant DNA technology are: process, isolation of DNA, use of restriction enzymes, ligation of DNA fragments into plasmids or vectors, introduction of plasmids or vectors into host cells. Step 1 is isolation of DNA. DNA is isolated from a source organism using various techniques, such as extraction from tissues or cells. Step 2 is use if restrictions enzyme. Restriction enzymes are used to cut DNA at specific sites, creating fragments of DNA. Step 3. Ligation. DNA fragments are joined together and inserted into a plasmid or vector, which acts as a carrier for the DNA and allows it to be introduced into host cells. Step 4 is introduction of plasmid. The plasmid or vector carrying the recombinant DNA is introduced into host cells, where it can replicate and express the new genetic material. Look at here the image. We take plasmid DNA. Then use restriction enzyme to cut it to 2 fragments. Then ligate the trans DNA into the plasmid, and the end chimera or recombinant DNA forms. Conclusion. Recombinant DNA technology is an important tool in biotechnology and genetic engineering, with numerous applications in medicine, agriculture, and industry. Ongoing research and development in this field is likely to lead to further advances and new applications in the future.